Now, let me introduce you our speaker today, Elena Martínez, Group Leader of the Biomimetic Systems for Cell Engineering Group of IBEC. Elena holds a PhD from the University of Barcelona. During her PhD, she was visiting student at the University of California in Berkeley and at the University of Uppsala. Afterwards, she did a postdoctoral stage at the EPFL in Lausanne, and in 2009, she was visiting researchers at the Imperial College of London with Professor Molly Stevens. In 2003, she received a Ramon y Cajal grant. In 2008, she became senior researcher within the group of Josep Samitier of IBEC. In 2013, she started as junior group leader of the Biomimetic Systems for Cell Engineering Group and was promoted in 2018 to senior group leader. From October, she is also professor at the University of Barcelona. She has an ERC consolidator and a proof of concept grants and coordinates a FET Open Consortium. Please, Elena, thank you for being here, and the floor is yours.
First of all, good morning to everyone, and thank you very much for joining this IBEC seminar. So what I will be trying to do today is trying to tell you a little bit what we have been doing in these last years, using biofabrication techniques towards writing cellular self-organization with the aim of developing models, biomimetic models of intestinal tissue. So let me just start by reminding you a little bit about the small intestinal epithelium, which is the tissue we are interested in. So this is a tissue which is the main responsible for the nutrient absorption, and also it forms a barrier that protects us from pathogens. And this is because epithelium forms a selective tissue barrier. Thanks to that, the cells are joined, forming tight junctions, which are just responsible for the paracellular transport of substances. So the tissue itself has a very interesting three-dimensional architecture with the formation, some villi and crypt microstructures. So those are structures that can go up to 1 millimeter in height and 200 millimeters in diameter. And the first role of these structures is to increase the surface area for the absorption of the tissue, but it also has some functionality in increasing the residence time of some drugs, for instance. Also, it creates an oxygen gradient for the microbiota. And what is also very interesting from this tissue is that the tissue that contains a lot of different types of cells, so a multicellular population. So we are interested in creating in vitro models of this tissue. And why do we want to use them for? We have several interests, including food testing, drug discovery, modeling diseases, toxicity studies, and drug testing. And of course, also, we would like to use them for basic studies. So up to now, the standard goal in vitro models are the monolayers, flat monolayers that are cultured on top of porous membranes, plastic porous membranes, and that they are using this system, which is transwell insert, and this system creates two chambers, and one that mimics the lumen in the apical side of the cells, and one that mimics the stroma in the basolateral side of the cells. So this is a very good format because it creates a way of culturing the cells in a standardized manner, and we have independent access to the apical and the basolateral sides. So, for instance, absorption tests can be performed during this in this model. However, the gold standard of this cell culture uses CACO2 cells that when they are cultured in this format, they are too tight, meaning that the barrier that they are creating makes that for instance, the uptake values of some substances, mainly slow absorbed drugs, they do not correlate with the in vivo data. So our aim was to improve this system, but creating what we call a biomimetic in vitro model of the small intestine. And here you have a nice cartoon of our ideal model, in which we want to keep some of the formats of the conventional model, for instance, this transwell system in which we have access to both the apical and the basolateral side. But we are interested in also having some other key issues of the in vivo tissue, such as, for instance, the multicellular population, the fact of having crypts and stems and villo-like structures, the fact of also having not only epithelial cells, but also other cell types such as fibroblasts or immune cells. So what I'm going to tell you is our efforts in trying to add to the basic system several of these components and what we have developed so far. So I will start by telling you how we have approached a way of including this multicellular population in our 3D model. And to do this, we are taking advantage of the multicellular composition of intestinal epithelial organoids to create what we call intestinal epithelial monolayers. So for those that are not familiar with the matter, intestinal epithelial organoids are formed by the culture of stem cells, which are residing at the bottom of the crypts of the intestinal tissue in three-dimensional matrices. The most popular is Matrigel. And then they, when these cells, they are placed within the matrix, they grow and they form these closed structures that recreate the morphology and the cellular composition of the crypt domains that are found in the tissue and the villus domains that are also found in the in vivo tissue. So when coming to, for instance, absorption tests with these organoids, the problem is that they are a structure which is reversed with respect to the one that we have in vivo, meaning that the lumen is an enclosed volume which is inside of a closed sphere. So the access is pretty much difficult. So our first task into this project was to try to develop a strategy in which we could transform a closed organoid into a monolayer, an open monolayer. To do that, what we have been working with is to play on the surface in which we culture these cells. So in collaboration with Eduard Valle from IRB, we are getting this mouse in which the intestinal stem cells are actually expressing a gene that makes them a reporter, a green fluorescent reporter. And then we are getting some intestinal crypts from this mouse. We are creating an organoid culture, and then from these organoids, we are getting either organoid-derived crypts or single cells. And then what we did was to culture these cells on a very thin film of Matrigel on a substrate, meaning we are presenting the cells with a hard substrate or with a thick layer of Matrigel on the same substrate. So we are presenting the cells with a soft environment. And what we found is that when we were placing the cells on a hard substrate, we had the formation of monolayers, intestinal epithelial monolayers. So the organoids were, the crypts, were forming these monolayers, either when pieces of crypts were seeded or when single cells were seeded. While when the substrate was keeping the soft environment, we didn't get these monolayers, but we just got organoids. Not only this, but then when we were looking at how these monolayers were being formed over the time, either when using crypt pieces or single cells, what we found, and this you can see the stem cells here in green, is that these cells, they were able to self-assemble and self-organize, creating some sort of crypt domains that were maintaining over the time while the cells were spreading and forming the monolayers. So you can see here these crypt villus domains that are preserved in the monolayers, while there is a population of cells that are not stem cells anymore and that we assimilate to villus-like domains. Then we wanted to use these cells, as I said, as a functional models. So it was very, it was very important for us that these cells, they could form an effective barrier that we could use, for instance, for testing the drug absorption or nutrient absorption. And then for that, we decided to try to do this, this culture on the standard transwell setups. And so we had, again, our porous, this time it was a porous membrane, a plastic porous membrane, and then we culture our cells on top of this system. And then these cells, they form monolayer. And we could see that the monolayers were preserving the population of some regions with proliferative cells, here marked by Ki67. We kept the crypt domains that are marked here with the stem cells in green with the GFP reporter. And they are also expressing FB2 receptor that it's also typical from the crypt domains. However, when doing this culture, what we found was that the monolayers were not able to cover the whole substrate. So we couldn't get an effective barrier from this cell culture system. Then we decided to go back to the biology of the organ, and looking at the biology of the crypt, we saw that actually it is known that the stem cells that are residing on the basis of the crypts, they are in contact with cell types which are called Paneth cells, that they provide them with a set of signals that are very important for their the maintenance of the cell niche or the proliferation. But also there is other cell types that are playing a predominant role in the formation of this stem cell niche, and those are, for instance, the intestinal subepithelial myofibroblasts that are just below the stem cells and the Paneth cells. And those are responsible for the production of a set of Wnt signals that are very important in the regulation and the proliferation of the stem cell niche. So with this in mind, what we did was actually trying to include this signal of non-epithelial origin in our system. So for that, what we did was to get from the intestinal tissue, not only the crypts, but also we continue the digestion of the tissue, and then we got this cell population that it was positively stained for the expression of alpha smooth muscle actin and vimentin, and it was negative for desmin. So this we considered as our population of intestinal subepithelial myofibroblasts. And then we culture these cells and we collect the supernatant that we called intestinal subepithelial myofibroblast condition medium. And then what we did was to include this condition medium in our cultures. And what we saw is that when we were including this signal in the culture, the epithelial monolayer was able to grow in a much more effective way. And we can see here the growth of the surface coverage over the time at 3 days of culture. And it was growing, forming a complete monolayer that covered the whole transwell area by day 10. And also it was preserved by day 20 of cell culture, where we, in both days, we thought we had a surface coverage of 100%. And this we could actually see because both at day 10 and day 20, we could measure one parameter, which is the resistance of the barrier that these cells were forming when being grown in the system. And we could see that this electrical resistance was actually increasing with the time of culture. And we also saw that the monolayer that was being formed here, it also preserved, it was a barrier for the passage of compounds that are used to model paracellular permeability, such as this FD4 dextran. We also saw that the monolayer that was formed, it was preserving a certain population of stem cells that over the time First, they proliferate quite a lot. So we had at day 10 quite a large population of proliferating cells. But then later on, this population of proliferative cells, they decreased. And we just had an increased population of differentiated cells. So to us, this indicates that the cells, they are differentiating in a proper manner over the time, keeping a pool of stem cells, so in such a way that the monolayer is kind of reaching an equilibrium, an homeostatic equilibrium in the system. So we could make this just by arranging the surface properties of our substrate. And then once we have this, we were wondering if we could actually use this strategy to, for instance, play with the positioning of the stem cells. And to do that, what we did was to challenge our system, trying to play with some ligands that are the EphrinB1 ligands. Sorry, these ligands, they are known to form gradient of expression over the height of the crypt. And this Ephrin gradient is the opposite as the one that is formed by the F receptor. And this has been seen in vivo, but it has not been reported in vitro. So we wanted to check if our monolayers that actually could create these domains, but in a non-regular manner, were able to respond to a surface in which this Ephrin ligand was placed in a micropattern manner. And this is work in progress, but what I can tell you from now is that first, to do this, we had to develop a method to actually create the micropatterns of these Ephrin ligands onto our Matrigel surfaces. So these surfaces are very soft, and it's not very easy to pattern them using conventional techniques, such as, for instance, microcontact printing. So to do that, we used a technique that we developed in the lab a few years ago, in which what we actually do is take the Matrigel in the gelated state and then freeze dry it, and then use microcontact printing to transfer the protein, and then later on reconstitute it again with the base solution. And so first of all, we just check that on these matrices that we have the Matrigel in a freeze-drying state, the cells, they could grow after reconstitution, and they preserve the same kind of self-organization that and cell population that we saw previously in the non-freezed substrates. And then once we saw that this was mostly equivalent to our first findings, then what we did was to try to pattern the Ephrin ligands. And here you can see a pattern of the ligands in stripes, and how these cells, they are growing on top of this pattern, and they are forming proliferative clusters. So these clusters, they are actually oriented in the shape by the stripes of the micropatterning, and they are also elongated in response to the surface micropattern that we are presenting to the cells. And not only this, but when we do some patterning of round areas in which we have the Ephrin ligand in some places, but not the Ephrin ligand in these round voids, we have that despite this is not, the pattern was not very successful, I would say, but we saw that first of all, the pitch interval that we were getting in this system, either these circles were separated 150 microns or 350 microns. This created aggregates that were proliferative aggregates, sorry, of the system that were approximately from the same size in both cases, but the distance by which these aggregates were separated was actually responding to the pitch of the substrate to which we were presenting the cells. So we could say that by this way, we could present the cells with some micropatterns of these EphrinB ligands, and we could guide by this system, by this in this manner, the cell organization in vitro, as it has been seen that it's guided in vivo. Then, once we have developed or played a little bit with a manner to create these intestinal epithelial monolayers, creating multicellularity, we were also wondering if we could take our monolayers and now provide them with a three-dimensional architecture. That is what we have in the native videos. And to do that, what we have been doing, is okay. So as a substrate, we decided to use hydrogels. Why? Because they are materials which have composition and mechanical properties and porous structure which is pretty similar to the extracellular matrix of soft tissues, mainly intestinal tissue. And then we decided to use hydrogels that could be polymerized by a free radical polymerization method, meaning that we have our polymer, and then we add a photoinitiator, and upon the UV light or visible light, this photoinitiator creates radicals, and this creates a polymeric 3D network. So this free radical polymerization system can be controlled spatiotemporally, and this is something that we like to be able to produce three-dimensional structures. It produces a system in which the polymerization is random, and it creates in the end a kind of heterogeneous networks. And then as a system to produce, to induce this polymerization, we kind of scan the light-based biofabrication strategies available. And there are some techniques that are based in additive manufacturing technologies, such as photolithography or digital-like processing. And then some techniques that are adapted to the biofabrication technologies, like again, lithography-based bioprinting or laser-assisted bioprinting. But then we decided to use something inspired in this digital light processing, but in a simple way, by adapting a photolithography method that it's actually the standard methodology to fabricate microstructures in the microelectronics industry. So as a hydrogel, as a polymeric compound, we decided to use polyethylene glycol diacrylate hydrogel, in which what we have done is just to create some pools of PDMS, and then include this PECTA polymer, and just use a 2D photomask with some open windows transparent to the UV light. So in such a way that when we shine the light on these structures, we could create these three-dimensional microstructures that are formed in the with the windows that are open to the passage of the light. Sorry. Okay, so as I said, this free radical polymerization has a beauty that can be specially controlled, and this is because it's a reaction that it's inhibited by the oxygen. So if we have in the system for the polymerization to take place, we have to deplete the oxygen of the system, and then once this is depleted, if we introduce an oxygen source, that the oxygen will be diffusing in the system, this will be able to control the polymerization. So if the ratio between the oxygen that is introduced into the system and the oxygen that is depleted is much smaller than 1, there will be no polymerization. If it is much larger than 1, there will be a fast polymerization. And if it is close to 1, we are in a situation in which we have an oxygen gradient along this vertical distance, and we can control where the polymerization is taking place. And this is what we have actually done. So when you can see that this time of diffusion actually depends on the dimension of the windows, on the open windows for the light passage in the system. So when we have no photomask, we basically have very high values of this number, and we have polymerization. But then when we have windows that have the range of microstructures, we are getting values of this parameter that are close to 1. So we are in a situation in which we have an oxygen gradient along this direction, and these structures with this roundish shape are formed just by doing nothing. So they are formed in a natural manner. And they can actually be controlled, I will show you in the morphology, by the intensity that we are shining into the system. So by this, we could produce this. Oops, I don't know why this is not working. Okay, I have some problems with my mouse. Okay, we can produce these villi-like structures. You have a top view here of the structures that are produced and a cross section of the same structures, and you can see this roundish morphology that it's pretty much in agreement with the morphology that is found in the villi of in our intestine. And because of the characteristics of the process, just by using the same mask, but just by changing some parameters such as the time exposure, we are able to control the dimensions of these structures. And not only this, but also we can control the mechanical properties of these structures as the gel fraction, so the amount of polymer that cross-links, is actually depending on the energy dose that we are shining into the system. So once before the saturation in this slope, we can control in here the mechanical properties that we are targeting just by controlling the parameters in which the radiation has been produced. And by this, we can achieve scaffolds that have stiffnesses which are in the range of a few kilopascals, and that are actually in the range that has been reported to be relevant for soft tissues. So the same technique also allows for a precise control of the gel functionalization. So to do that, what we do is we copolymerize our PECDA hydrogel with acrylic acid. And then on the groups of the acidic groups that we have created on the surface by EDC NHS coupling, we could incorporate a few, proteins. And here you can see a picture of just a model protein. This is streptavidin Texas red, but this is just to show you that the system is working. And upon functionalization with, for instance, collagen, what we can then see is that the intestinal epithelial cells, either primary cells or caco2 cells, they can grow and form a continuous monolayer on the surface of the scaffolds. And if we do cutting, histological cutting, and we observe the morphology of the cells that are covering these villi structures, we could see that these cells, here you can see the actin in red, they are looking very much to a monolayer of heavily polarized cells that are covering the villi-like structures. And if we look at some polarization markers, such as the presence of villi, that you can see here in green in the right position, or the presence of ZO1, which is actually staining the tight junctions of the cells and should be located in the apical site, and this is what we see here. And also beta-catenin, which is stained here in cyan, and it should be located at the basolateral site of the cells. All these markers, they tell us that the cells are polarized as they should be, and we are expecting to have in the intestinal epithelial cells. Not only this, but we know that in the in vivo tissue, this is an histological cutting of the in vivo tissue, the orientation of the cells, the shape, the morphology they have, and the package ratio depends on their position along these curved structures. And this is also what we have seen in our system. So these cells, they can have, depending on their position, first of all, they can have different height, that it's depending on their position along the villi structures, but also they have different morphology that you can see in here, and different orientation of the nucleus with respect to the hydrogel surface, that it's also changing along the villous vertical axis, as it does in vivo. So then, once we saw that the morphology of the monolayers was the proper one, we wanted to check if they were functional, as we want to use them for screening. And to do that, what we have done is we measure the conventional parameter that is usually measured in the system, which is the transepithelial electrical resistance. And what we have seen is that actually we get values of the transepithelial electrical resistance, which are much closer to the values that are reported for the human intestine than the ones that are usually obtained in conventional monolayers cultured on hard substrates, which are very far away from the physiological values. Not only this, but also we've seen that our 3D scaffolds, they are also much more permeable to the paracellular testing compounds. And this also corresponds to a liquor epithelium as it is in vivo. So we developed a model that can be used to perform this functional test, providing information that correlates in a better manner with the physiological information coming from the in vivo testing. However, we have, I don't know why this keeps happening, but however, as we said at the beginning, we wanted to develop something that resembles to this model that you see here. And then in here, we do not only have the epithelial component, but we also have some other compartments, such as the stromal compartment in here below, in which there are different cell types that have a very that are highly relevant for the functional of the intestinal epithelium. And among them, we have myofibroblasts, but we also have fibroblasts and immune cells. So I'm going to tell you what we have been working, trying to incorporate also these other cell types in our model. And then to do that, the first thing we had to do is to allow our hydrogel to be a good matrix to embed cells, and to allow cell growth and cell, for instance, cell extracellular matrix deposition and other functions within the bulk of the hydrogel. And then to do that, PECDA is not a very good material because it's synthetic, so non-biodegradable and completely inert network. So we have been working on developing these co-networks in which to the PECDA initial polymer we had, we are also adding gelatin methacrylate material. So we combine both with a photoinitiator and then light. In this case, we filter the UV light just to 365 to minimize cell damage. And then we create these co-networks that now they can be degradable. So we can see here that they are degraded with the time upon addition of collagenase. And depending on the composition, so the fraction, the proportion that we have from from the PECDA compound or from the GelMA compound, this degradation rate can be tuned. Also, the mechanical properties can be tuned depending on the total macromen content of the polymer, and also depending on the composition of the co-networks. So, taking into account these properties, what we have done is to create a model in which we have not only the epithelial component, the CACO2 monolayers, but also an stromal compartment in which we have embedded fibroblasts. So in this model, what we have done is we have a prepolymer solution in which we have the our fibroblasts, and then we polymerize the system, getting some samples upon which we mount it on transwells, and we grow the CACO2 monolayers. And this is, we leave the system growing in here for 21 days. And then upon 21 days, we just did some cross-sectional cutting, histological cutting, and staining of the system. And we could identify the CACO2 monolayers that were growing here and the fibroblasts that were embedded within the structure. And this you can see nicely both compartments, and you can see that the epithelial cells, they have the expression of actin in the proper way, and beta-catenin. And we could also see that the fibroblasts that were embedded within the matrix, they were able to synthesize collagen 4, and we actually saw accumulation of collagen 4 at the basement membrane, separating the epithelial layer from the stromal compartment. Then we check if the presence of this layer was affecting the properties of the epithelium as a monolayer, because we know that this happens in vivo. And well, first of all, we saw that actually the morphology of the epithelial layer was changing over the time. So this is at day 8, and this is the sample that doesn't have the fibroblast, and this is the sample that has the fibroblast. And then we could see that actually the epithelial monolayer formed in the presence of the fibroblast, it has a much better morphology than the one formed without the presence of the fibroblast. But not only that, when checking at the formation of the monolayer, what we saw by measuring the transepithelial electrical resistance, we saw that the presence of the fibroblast was in making the monolayer to be formed faster with the time. This is this graph that you see here, and also achieving higher values of the TR. This was also reflected on the permeability of the monolayers formed. And then what we did was a test to check which could be the role of these fibroblasts when there was in restoring the barrier formation of the epithelial layer when there was a damage produced. So what we did is to produce some damage in the epithelial layer and then check how was this recovery of the TR value. And we saw that this recovery, the slope of the recovery was much enhanced when we have this fibroblasts in the system. Okay, so this looked like a very nice material, but also it has the benefit that we could actually tune some more properties, such as, for instance, we could tune the cross-linking degree of the material. And we could do that just by changing the methacrylation degree of the GelMA that we are using. So to methacrylate, to get gelatin methacrylated, we have to mix gelatin with methacrylic anhydride. And then depending on the amount of this compound that you add into the reaction, you can have a material that has a high methacrylation degree or a low methacrylation degree. And depending on this, we can tune, for instance, the biodegradability of the material. So we can make a material that is much more degradable by just decreasing the methacrylation degree. And we saw that even in this case, so with a material that has a less methacrylation degree, the CACO2 cells were able to grow on top of the material and form a nice monolayer that has very nice TR values. And what we actually saw was that by employing this strategy of having a material that is much more degradable, we could get these fibroblasts that we have been embedding into the stromal compartment to get them not in a round shape, but more like a spread-like shape. And they were actually able to migrate to the structure, and they were located just underneath the epithelial monolayer, as they are supposed to be located in vivo. And we can see here in a zoom how these fibroblasts, they are expressing collagen, and how this collagen is actually accumulated at the interface between the stromal compartment and the epithelial layer. Not only this, but then in this model, what we had tried was then to use a more relevant cells for the in vivo composition. And then we embedded some intestinal myofibroblasts and some macrophages. And this is what you can see in here. So all these cells are a mixture of these intestinal fibroblasts and macrophages. But what we have was that these roundish cell types that are kind of clustered, they correspond to actually the immune cells that were embedded into the system, while these other cells that were expressing collagen in here, they correspond to the intestinal myofibroblasts. And then by including these immune cells into the system, what we could see was that, first of all, the epithelial monolayer was able to be formed the same way as if we didn't have the immune compartment. But then we could damage the system, so we could produce by LPS treatment a disruption of the tight junctions, and then we could see how the system was responding in the presence of the immune cells or without the presence of the immune cells. And to do that, we checked two interleukins, interleukin 8 and interleukin 6, which are indicative of an inflammatory state. And we saw that, especially for the interleukin 8, we had a lot of expression and secretion of this cytokine when we have added the macrophages on the system. So this means that we have been developing with the model an inflammatory in vitro model of the intestinal mucosa. So then we said, if we have now like this model in which we have the stromal compartment and the epithelial compartment, can we now put it together with our three-dimensional structure and create an overall structure with this shape? So to do that, we tried to use the photolithography strategy that we did without the presence, so with just the PECTA material, without the GelMA. But then we found that we didn't have much success by using just photolithography. And then we explored other options. And other options, we came back to these solutions that the existing techniques in the light biofabrication arena. And we decided to use a variation of this digital light processing system, but including in this resist, in which usually this is not the case, cells in the formulation. So trying to do something that will mix up this biofabrication, but with the DLP techniques. And to do that, what we have been using is a very low cross printing system. This is actually the system that is located in the microfab facility of IBEC. And the system works in such a way. So we have a bath in which there is a window which is transparent. And we have a light source that is a projector. And we have a support in which we the biofabricated structure grows. So basically, we have a CAD design that we are introducing into the computer, and then there will be some polymerization layer by layer in here of this CAD design, and we will obtain the polymerized structure in this support that is moving upwards during the printing process. So in principle, the resolution of the system is about 50 microns. And what we have been doing with the system to be able to use it for bioprinting is customize the support and the bath, including an IR filter, infrared filter, to be able to avoid heating of the cells, and also including a thermostat. So everything is heated up for 37 degrees. And this system, they can shine with a power range between 3 and 12 milliwatts centimeter square. So basically, the X and Y resolution of the system is controlled by the projection, so we cannot do much. But the zeta resolution of the system, so the thickness of this layer, It's actually controlled by what we call the curing depth, because of this transparent window, there is a layer here that it's not polymerized. And when we add here a bioink, we have a curing depth that depends on this parameter, that depends on the bioink properties, but also on the dose of the intensity that we are providing with the system. So basically, if the intensity is higher than this EC parameter, there will be polymerization. If not, there will not be polymerization. And then to control these properties, as I said, this depends on the bioink properties. And we use as bioink something that it's composed by a polymer. In our case, it's a mixture of PECDA and GelMA, because we already know that the cells, they were liking these both polymers. As a photoinitiator, we now use LAP, which is has an absorption peak in the visible range. And we also add to the system other elements that are also important for polymerization, that are oxygen, to control polymerization, that are oxygen scavengers, and photoabsorbers. In such a way that this is the process. So we have our bioink preparation, to which we could add cells. Then we have the CAD model, and we can print on the substrate. And the beauty of this is that we can actually print on these substrates that we could, they can be glass substrates, but also polymer membranes. And we can just take this and either put it in a well plate, or directly incorporate it into a transwell device. So it's very easy then to adapt this to a well plate format to perform high throughput experiments. Then this is the type of structures that we could generate. So this is the CAD design with in this case, the crypt diameters and villi, the crypt structures and the villi structures. And this is what you can see here. So I don't know if you could see the crypts. They are barely seen in here, but you can see them in this other picture, in which we are focusing through different planes of this structure. And in here, there are cells grown. So cockroach cells that have been cultured for 21 days on the scaffolds. And you could see the cells growing at the tip of the villi structures, and also growing at the crypts of the structures. And then finally, we've used this system to create this mucosa models, in which we could bioprint the fibroblasts, and then on top of the system, culture the CACO2 epithelial cells. So unfortunately here, when we did the cutting of this system, the pillars that were on the surface, they just fall down. So you cannot see very nice the shape, just a pump. But you can see the fibroblasts, and how these fibroblasts, they have this migratory phenotype, and they are located and interacting with the epithelial cells. And these epithelial cells, they are expressing beta-catenin. The fibroblasts, they are secreting collagen. That is what they should do. And the cells look nicely polarized, with the actin on mostly expressed on the apical side of the neck. So this is pretty much the end of my talk. This is what we are here, and we are intended to use this system now to print not only one sample one by one, but in the new project, we will print just the whole plate in a single printing procedure. So I hope I could I've shown you an overview of the activities that we are doing in the lab towards this formation of 3D models of epithelium. And I want to conclude just by thanking our main partners in this project, that are Professor Eduard Valle from IRB, Dr. Samuel Ojos Negros from IBEC, and Pablo Loza at the IFO. And then, of course, the founders, but also the team. I'm not going to mention anyone, because all of them, they have contributed to the work I've presented here. And of course, I will be open for questions.